well?
I'm trying, but
Live streaming is on.
Okay.
it looks like it's either one or the other, right?
Okay, well, let's do with your slides, and then for the questions, we can open the cameras.
Well, I
And maybe let's do some. Yeah.
in my desk for nothing then.
Okay.
That's fine. I think that should do it.
Excellent. And to the online participants, let's make a competition of how many cameras we can have on during the session. That would be great. So we have a feeling of people on site at the same time. So if you are able to share your camera, all the participants, not only the speakers, but all the participants, that will be great. Maybe we will test the capacity of Dryfta, but this would be nice to see you in person, let's say virtually. Great. So we will start the session 3 called Microbial Processes in the Water Chain. So far, what did we learn? We have learned about microbial ecology and water engineering and water processes. We dig into the microbial processes this afternoon more for biorefinery approaches. And now what happens in the water chain, and there is a strong link here with drinking water. So this session is essentially about drinking water, but making links with treatment systems, with groundwater, with distribution, with analytical methods that we can apply as well, and the link with groundwater even. So we have the pleasure to have Professor Michel Prévost from Polytechnique Montréal in Quebec, Canada, who is the chair holder of the Industrial Research Chair for Drinking Water from NSERC or CRSNG in Canada. And she will speak about how can we use microbial ecology to identify sustainable, we have an echo, treatment solution in water systems.
Okay.
Could check because there is a problem in our room about listening, and
David,
okay.
is it okay to start?
Yes, you can start, Michel. Thank you. Floor is yours.
Thank you. So first of all, I'd like to thank the organizing committee and the chairs from this session for inviting me to share my views on how methods and approaches from microbial ecology or microbial aquatic ecology really can be used to identify sustainable treatment solutions in drinking water systems. First of all, I'll share how I got to these microbial ecology methods. As a student a long time ago, I've been at this for a while. I worked on biological filters, activated carbon filters in drinking water treatment plants, and I was looking for ways to measure the biomass and understand which media were better for such filters. So I had questions that I had to bring to Dr. Pierre Servet that you see on the right-hand side here. Pierre became a friend and was very, very generous with his knowledge and his models he had then proposed for biological filters and drinking water distribution systems to answer things like how many organisms, how many predators, bacteria, how much biomass, what's the activity, the potential activity, the real activity, what design parameters can be used, and how much of an impact would they have on fixed and suspended biomass, and actually can it work in Canada with temperatures of 1 degree C in water for 4 or 5 months a year, and finally can it be modeled so we can use it for design. And as I said, Pierre was generous with his friendship and his knowledge, and unfortunately, as he left us today, I every time I go to the lab, I kind of think of how much I owe to him and how much I will miss him. But we were looking specifically at biofilm and suspended organism, looking at what we call classical methods now, like live dead epi, staining, density and activity using labeled substrates, fish probes, and relating those to environmental parameters, process operation, and the characterization of substrates to predict performance and determine design of these filters. So when you look at solving engineering problems now. In the last 15 years, there's an enormous amount of new tools. I can hear there's another microphone that's open, so that'd be nice if it be closed. And these tools provide faster, in some cases, not in others, but certainly more informative responses. And we can think about quick and selective enumeration of cells or biomass density. I know Dr. Freud will talk about online flow cytometry later on in this session. What communities are present, what are they capable of, and actually what's most important, what are they actually doing? And as we use these methods, and some of which are not that quick return type methods, we want to answer questions about the processes and most importantly, actually understand what determines the communities that are present and what is our ability in process engineering to select the most resilient communities for the target process or environmental outcomes we desire, predict the impact of treatment on the communities, and most of all, in my case, I've been working quite a bit to prevent the presence of problem organisms such as pathogens. So really a high level view to think about what is it we can get out of a combination of several of these methods when we do these investigations. And when you solve engineering challenges, you need to do it at several scales of experimentation and final validation. And those include laboratory, very well-controlled experimentation, bench scale testing, fixed and mobile treatment plants, online monitoring, and real system validation. I find, especially in building water systems, that the reality is much more complex than any clever pilot we can devise. And I will bring a couple of examples with classical and new methods of I hope to make you think about the challenges of understanding at the microscale. What I call the microscale is you'll see in a building a tap versus a whole system versus the whole distribution system from the treatment plant. And thinking about how to sample to obtain a representative sample, how often to sample to capture the dynamics of the system, and what to measure. And these are key questions. Every time I get into a study, there are limitations in time, in resources, and our ability to have something representative of the system that allows us for predictive capacity is often limited. So I'll share with you a couple of my recent experience on how water systems affect diversity, hopefully making you think about a couple of questions that in each of your exciting studies. So just as background, Legionella has become a foremost pathogen, waterborne pathogen. The 2019 National Academy of Science, Engineering, and Medicine report on Legionella management summarizes it very nicely. The CDC estimates are that they cost about $1 billion a year. It is no longer driven, the cost of waterborne disease, by fecal pathogen, but mainly by Legionnaire's disease, otitis externa, and NTMs. So if you look at the table on the left-hand side here, you can see that the annual cost of the hospitalization for crypto and giardia type infections have gone down quite a bit, whereas Legionnaire's disease, which is underestimated at this time, is really an important component of that. The other thing you can see on the right-hand side that shows the etiology of drinking water-associated outbreaks is that there's this black showing up later here, and the latest numbers are even higher. The black is Legionella, and you can see that it takes a greater and greater share in the US and in Canada of the disease associated with water. So why is it so interesting and so complex to control Legionella? Because the built environment is a major ecological niche for the bacteria and controls focused on biocides for cooling tower and hospitals. We have very little ways and approaches and design criteria and building codes that actually address that well. And again, in the NASM report, we worked very hard to summarize the main control strategies and their limitations. And we came out with clear evidence of the influence of green building and water and energy conservation, the trade-off there. If you want to save water and save energy, this means you use less water and you don't heat up the water as much. And those are really important control strategies for Legionella, for example. And two other things came out and are really relevant for this group. There's a big future for prebiotic and probiotic approaches of control, and I'll talk about that a little bit in my talk. And we have to think about other opportunistic pathogens in buildings. We can't just have this one focus on Legionella and forget about, for example, Pseudomonas aeruginosa or NTMs. So building water systems are complex. They're complex in terms of devices, materials, hydraulics, water demand, temperature, nutrients, oxidants. I did most of my research in plants and big systems, and I can tell you building water systems are so much more, they're a miniature complex system compared to the larger distribution systems. And if you look at these pictures, you can see how not only materials in terms of the main material, but the accumulation of corrosion, or here the deposits here on the surface of a plaque from a heat exchanger can be so important. And when you look at a very simple hot water system, so we're really talking about just one of the two water systems or three or four water systems in a building, you have this main loop that is really bringing out the water and bringing it back through the main recirculation loop. And then you have these, and that's really simple hydraulics. You can predict what happens there and you can control things pretty easily. Then you have what we call, the British call subordinate flow and return loops, which are basically the secondary more branches of your system. And those are way more complex. You see two architecture here. And what is most important here, so you have water going up, water going back, and you have this, so you're really looping this system so it circulates if it works well. But then you have this little bit here, which can be anywhere between just a few meters to just a few centimeters. It really depends on the local system, but it is in this system what I will refer to in my future slides as distal sections of a system that a lot of things happen. So bear that in mind: primary, secondary, distal. So just to decide where you want to sample. For example, if you sample after flushing, which may be really most of the regulations until recently were in semi-flushed samples. Still, Germany, for example, has semi-flushed samples for Legionella. This here shows, and those are that's not Legionella, those are HPCs, those are culturable, and this is Pseudomonas aeruginosa. And we did these studies to show as you sequentially sample up to 5 liters. So this is not a big volume. This is like really 15 ml. And you measure in hot and cold water, hot, red, blue, cold. And you see the numbers go down drastically. Now, may drive, actually, we've shown
Live streaming is on.
It actually will drive the risk. And these are important things to consider when you start sampling a system. Again, when you decide where to sample in a large hospital like the one on the left that I'm showing here, where we worked on an outbreak investigation, if you select your points of sampling for example here, depending on the temperature at first draw, you will find that the ones that do not reach high temperature, even after some flushing, will be much more prone to have a high percentage of positive and to have higher concentrations of positive for pneumophila. This is important because depending on how you select your sampling points, you may or may not find Legionella, whether it is by culture, PCR, or other methods. So this is about 2 log difference in terms of readings that are seen. So it would give you quite a bit of a different perspective depending on where to sample. So one other question that you want to ask is, when we talk about diversity, we also talk about diversity in strains of Legionella pneumophila. And one of the questions we had was, first of all, is there a lot of diversity for these strains in a hot water system? And would it be different, the conditions in that distal part, would it lead to different strains or more and different strains to persist at these sites? And here you can see the results from a healthcare facilities in which we processed 32 positive samples, 47 isolates, and we looked at STs, SBTs in there to look at the diversity, found two dominant sequence type shown by colors here, the gray being unknown, but very like ST378. And as you can see here on the top here, you have, this is again the approach that we take for sequential sampling. So 0.5 liter and then 1 and 2 liters. And you can see that you will find those are flush samples here. And you see that you find both STs in flushed water and at first draw, but you don't find them. If you go 2 days in a row, you may find one on one day and the other one on another day. This has really important implication when you're looking for the identification of a source, for example, for nosocomial cases. So let's move on to unintended consequences of energy conservation. This is important because we all want to lower water usage. We all want to conserve energy. We have to go there. So how can we do that? And are there things to watch for? So I'll give you an example on how it is important to be careful of these trade-offs between energy, water conservation, and water quality. This is a study in a regional bed of 400 beds with 2 Legionnaires' disease cases in one wing and clinical samplings. And the hospital just had received a $4 million grant for energy conservation. And the system temperature was lowered, and new heat exchangers for energy conservation were installed just a few months before the outbreak. They actually won a very nice award for this, and everybody was very proud of their initiative. And then 18 months after this installation, we found in a study that there was a lot of Legionella after the first case. And basically, in the 2 wings, 88 and 56% of the samples were positive at the time by culture, which is the standard reference method for public health. So hot water systems were highly contaminated in both wings. So we kind of looked at, okay, let's find out what happens in the system. And to what extent can we find, is it going down to 55 that did this? Is it the installation of the heat exchanger? And you can see on the left-hand side here what a heat exchanger looks like. Basically, it's multiplying a surface. The surface present in this heat exchanger is equivalent to the surface in the whole rest of the building. So this is a very large surface. It's on purpose. It's a heat exchanger. And the temperature comes in at 20 degrees. That's the example during the period we sample. And it comes out around 45. So basically it stagnates about half the time. At nighttime, there's not much makeup water needed. So it stagnates, temperature varies then. So you can see that these are conditions that are prone to the growth of Legionella, especially in biofilm. You can see how deposits accumulate in such heat exchanger, and on the right-hand side you can see the results of Qpcr and culture-based investigations of densities with swabs. And more importantly, what was shown is that the epi strain that caused the nosocomial cases and was the same as had been isolated in the last 15 years, but at very low levels in that hospital. But because of this heat exchanger, effect, the concentrations exceeded the levels that could represent a significant risk to patients. So that heat exchanger was put offline, is operated differently, and with the matching of the clinical and the environmental strains was shown to be a aggravating factor in this outbreak. Now, let's move on to other methods that I use as well. I've not brought a lot of data to show what flow cytometry can do, and I'm showing here some of the partial results from a pilot study bench scale study that I finished last year, and we're working the data right now in the diversity data, which is quite exciting. I just wanted to show that these tools are wonderful. One of the things we looked at in this study was the seeding with host, and the difficulty we had was to be able to enumerate the cysts after the modification of temperature, for example, and we use live dead staining on this and flow cytometry, and it could show quite nice results in being able to follow the kill of our Vermabeba during these bench scale studies. And you can see two of the postdoc and PhD student, to Margot Duberva and Margot Casal, that are worked on the study in the picture. So I'll move on now to examples of recommissioning buildings after extended COVID. And I noted that Dr. Cheng will be presenting some extended stagnation data in this session as well. I'll be using some information from our applications of the framework that was published by AWWA and the regional guidance that was actually the directive that was issued by our Quebec government as well. And the idea behind these framework is to provide a decision-making framework for building managers to respond to building water system stagnation, and really help them rapidly during the pandemic, whether they had or not a water management program. And the main actions in these, most of these guidance, and there are literally more than 50 guidance right now, that I can think of rapidly, and most of them rely on flushing and or disinfection. I'll show you some results on flushing. Disinfection work is still going on for us. So this recommissioning flushing study was done in a sports complex. Obviously, it was closed during pandemic. And here you can see a very nice Olympic sports complex, and you can see the multiple showers just before going into the pool, for example. And those were, it's a single hot water recirculation loop, and then it goes out to single thermostatic mixing valves that feed a group of up to 22 showers. And I'll be showing you just a little bit of the data, looking at first draw and 5-minute flushing. So again, the distal part, and after 5 minutes, we're really out, and we're in the main loop of that system. So if we look at the impact of stagnation after 4 months of stagnation, so that was after 4 months of closure, then we did this flushing, this intense flushing for each point as prescribed by our local guidance, and then let it stand more or less, because there was very little use for 1 month, and went back to see if the benefit of systematic flushing of every single shower made a difference. So I'm showing results for two parameters here. The MPN, which is the lead alert based equivalent of culture, faster and much easier to perform in an engineering lab. And on the right-hand side, the LP Qpcr results. And there are two colors in these graphs. These are levels in concentration. GUs are MPNs. And the dark gray being the first draw, and the 5-minute flush, which is prescribed at each shower, is the light gray. And if you look at for the culture base, you can see, first of all, that the first flush numbers are quite high, over 10 to the fourth per liter. And the flushed samples after extended stagnation are quite high. Immediately after flushing, 1 day, these numbers are down by like almost 2 orders of magnitude, but they do bounce up, not as high as before. And that is one single flushing event to numbers. And again, there's a difference. It is higher with the first draw than the second draw. Now, if you look at when you see results with LP by Qpcr, the trends are different. Yes, the second draw is lower, but the difference between the two is less pronounced, which is quite important in terms of understanding what happens in there. And the other thing I'd like to point out, which is really important, is that there is quite a wide range of values obtained from different points of use in the same building that was operated under the same conditions. So this shows two things. It shows that the distal contamination is higher than the main system, and it also shows that it is quite variable from one point of use to the next one. And at the end of the day, you want to know what you use these numbers for, and you have to bring it back into risk. And using QMRA, you can look at different risks after different intervention and different stagnation time and bring that back to actionable items. That center was just reopened 2 weeks ago after installing 15-minute per day flushes at each of these group showers facilities, and with more than 90% non-detect either by culture and very, very low numbers by Qpcr. So flushing can work if it's managed well. And one last set of data on the water side, which is looking at the impact of water treatment regimes on Legionella in cooling towers. And as you can see here, for different cooling towers monitored over a year by 16S and 18S, here you can see the bacterial genera, and you can see there's quite a bit of variability in this
Excuse me, Michel.
Yeah, il reste 3 minutes. Hein, si j'ai bien
Non, ce n'est pas le problème. Le problème, c'est qu'il y a un problème d'écran. There is a problem with the screen, and nobody can see your screen anymore. It's not your problem, so please stay as you are, and we are trying to solve where is the problem.
Oh, wow, I can see. It's a blank screen now. Okay.
No, but please stay as you were with your presentation, because the problem is between...
It's on your side?
Yeah, and the room here, the video, and because there is also a parallel.
Yeah, there were 2 messages showing that. Actually, I had 2 messages showing here. I heard 2 messages that it was ongoing, not ongoing anymore for sharing. I thought it was perhaps somebody else, but I guess it was my screen, not sharing.
No problem from your direction. So everything works fine from your direction. It's more that there are 2 parallel streamings. And there is a problem, so we just hold the time. I'm sorry for this inconvenience to everybody, but...
okay, so what shall I do, David?
We just wait, so let's just hold the time. And our colleagues from the technical part are checking. Sorry for that.
Oh, wow, I can see. Oh, I can see the... parallel now, which you can... Okay, so somebody shared the slide. Thank you, Julia.
And also Zeb from Dryfta, please make sure that it works in YouTube. We cannot afford that we have 220 participants overseas that cannot see the presentations. That's ultra capital that everything works on the platform.
Perhaps it's... Oh, wow, it's so cool to see everybody's face. But
that we managed.
Yeah, that part works well. I can see you from a remote camera now. It looks cool. Oh man, that's so cool. It's a big group.
And from the Senate salle, were you able to call Eric? Is it Daphne, the name of the colleague? But there is a problem here as well because we don't see the presentation now. Oh. Can you try to share your screen again, Michelle, please?
I will.
Or your presentation, sorry. Yeah.
Yeah.
We see you.
I'll do that. Okay, I'll do that.
I think it's stopped.
Voila.
And now you have to press again the same button.
Yeah.
Sorry.
Whoops. It won't take it this time. Just okay. Now okay. I just gave it an authorization, so now it's working.
So now we see your desktop, and you should launch your presentation in PowerPoint now.
Do you think it's okay now to start again, or?
Yeah, can you just click on it now, just to see how it looks like for everybody? Yes, so we see your slide. Amit, do you see the slides of Michel? Yeah, so it works. And in parallel, in YouTube, does it work? Yes. Yeah, so everything works again. So we can continue. Thanks. My apologies for all these issues that we have.
Yeah, and I realize I only have about 5 minutes left, so I'll be quick.
No, but we have plenty of time. We have buffer time with the breakout session. So please go at your rhythm that you need to come. So there is no problem.
Okay. So again, let's move on to cooling towers. That's a project with Dr. Fauché from McGill University. And here you show the bacterial genera in cooling towers at different dates, monitored over a year. And the objective of this first study that was recently published was to characterize the bacterial eukaryotic. Man, this is a terrible word for a French girl. Eukaryotic is fine, but communities in these cooling towers to understand which communities are not favorable to Legionella. So we know that for those of you who don't work in Legionella, that without any hosts, there's very little growth, significant growth that could lead to high enough numbers or concentrations of Legionella that could cause a significant risk. That is why Kiran worked with Dr. Fauché to analyze this data from the 18 towers and just recently published a result showing that, as expected, a very complex ecosystem with primary producers, grazers, and several functional bacterial groups, but more importantly, several taxa harboring known as Legionella pneumophila host species, and well-established host cell species such as Acanthamoeba, Vermamoeba, Naegleria, and Tetrahyena. So to us, it really drives the project that I'm leading right now to understand if the treatment that we're imposing on these towers should be aiming those hosts rather than Legionella, which is more or less a consequence of the opportunity of these hosts to harbor Legionella. And an interesting twist that Kiran shown was the fact that Brevundimonas was able to stimulate the growth of Legionella. So again, showing the importance of community interactions. And also shown that the diversity were molded. So the communities we found in these towers all across Quebec were molded by the quality of water that fed the makeup water, which was kind of a surprise for us and is a challenge to understand at this point because some of them are not that different. And we also showed I think the first actionable information bit that came out of this study for the operation of cooling towers with that. There was quite an impact on the weekly, daily, and continuous modes of operation in terms of chlorination. So weekly means once a week you add the disinfectant, whereas continuous, it's adjusted to maintain an oxidative pressure regular throughout the week. And so that is my last example for you. I'm just closing participating in closing a large project called ATTRAP, which is Algal Bloom Treatment Risk Assessment Prediction and Prevention through Genomics, working with the University of Montreal, Dr. Shapiro and Dr. Sauvé. And looking, and my part of it was to work and understand what these tools could, how these tools could help us identify the critical points of accumulation in the water system, how we can treat the cyanobacterial blooms in water and sludge, and how we can better manage these accumulation in water treatment plants. So I'll give you two examples. First is defining the design criteria for the oxidation of algal blooms. I worked for about 10 years looking at lab-based studies to understand whether pre-oxidation could be a solution to prevent the accumulation in sludge and then prevent the issues with the disposal and treatment of that sludge or recirculation of the supernatant of the sludge. And Saber just finished his PhD just a few months ago and published a suite of studies, some lab, some full scale, showing on model organism which were the impact of different oxidants such as ozone, chlorine, and peroxide in terms of the physical structure, the release of the toxins, and the different spectra of the different cells. And the last part, for which we're still working on the two last papers with Farhad Jalili, the PhD student, is looking at what happens in the sludge. When you remove these cells from water using conventional or membrane systems, you end up with sludge. These sludge are laden with cyanobacterial cells and toxins. And the first thing that we looked at with Farhad is how constant this is, how this diversity varies throughout a year, reflecting what comes in and is being captured by the separation processes. But most importantly, what happens when you have these sludges stagnate or be kept in a sludge holding tank, especially for small utilities. That's what they do. They just pile up the sludge for a while and then try to do something with it. And how important would be stagnation? Would we have a degradation of the toxins with the biomass present? Would we have a survival? Would we even have growth, which had been suggested in some studies? And we did show that there was a clear shift in the communities from the sensitive genus such as Dolicho to the more resistant genus such as Microcystis during stagnation for 8 to 30 days. We also showed significant growth, which was really surprising by traditional cell counts accompanying the stagnation shifts that we saw with the metagenomics. And we also showed that if you use this combination of shotgun metagenomics, stacked cell counts, cyanotoxins, and ddPCR, we got consistent and complementary evidence to show that there are things that should be done to prevent the growth and the toxin production during storage. That has significant direct implications for management. So my closing comments is these in-depth investigation of diversity and function are extraordinarily interesting and challenging, but they're also very resource consuming because there's a limited number of samples in time that can be processed. And that is a real challenge to the study of complex and dynamic system, and snapshot in time it may not be enough to understand it. It makes for a lot of insight and understanding of the system, but for a dynamic system there are limitations to that, and hopefully in the future it will be easier with some of these tools to get answers more quickly. Understanding the dynamics, defining the diversity in biofilm and water. In water systems, to me, the detachment part is poorly defined, and we look at the communities in water and the communities in biofilm. We can't really understand what goes on between the two and the rate of detachment versus growth. these are not steady state systems. The water demand in the system changes all the time, depending on the user's demand profile. So this is, to me, something that is really important for the future, and hopefully all of these tools will help us understand this. And we need to turn this incredible information to actionable results to understand and design engineering system. We have made a lot of progress, but there's still a lot of work to do to show how this can be made. And finally, we need to bridge this new information to risk assessment. At this point, the health community is very conservative in using culture as the gold standard for risk assessment. The NASM report also confirmed that. But we need to move and push this so these new methods are now used to assess risk on a more systematic manner. So I'll thank you for your attention, and I'd like to recognize these are recent commissioning teams, pictures, and I'd like to acknowledge the work of all my nice students and collaborators here at Polytechnique, which I did not name one by one, and they are key to the success of what we do. So thank you for your attention.
Warmest acknowledgement, Michel, for your presentation, and it's very dynamic as well. It's much appreciated, and it's impressive the different topics that you have covered in this keynote across water systems and drinking water line. There are some claps you see from the participants.
It's cool.
Really appreciated. I think we should do more conferences without slides to make it a bit more ex cathedra, like in universities in the past. But it seems that you gave really nice messages. Thank you so much for this highlight from Montreal.
Thank you.
Are there questions from maybe the drinking water specialists in the room or online? In the chat, Sophie and Sergio, can you look at if there are some questions in the Q&A?
We have two questions from Dr. Claudia. So the first one, what do you think about, or do you want to ask yourself? Okay. What do you think about the difference between cultural methods and Qpcr methods to detect pathogens?
Well, as everybody else, Qpcr and dpcr are quick. So, we're all looking for quick methods to confirm. And as everybody else, we look at viable Qpcr for a while, dabbled in that, and looked at the very nice work done in Europe on it. And saw some different results in terms of success. So when you ask me what do I think about between culture and Qpcr, I use Qpcr for process control. I base all of my assessment of a water system on Qpcr. I get it the next day. I know how much there is, but I cannot go to public health with a Qpcr. There is no opening to consider 200,000 per liter Qpcr as a valid result, with few exceptions in terms of risk. So I think we need to bridge those two. There are limitations to culture, which we all are aware, that are being put forward more and more to show that culture has serious limitations to predict infection. But there are also very recent information from the University of Vienna that says that no, Qpcr is not in any way indicative of risk. So for my applications, as I said, I use it as the process control in cooling towers and hot water systems, but I cannot use it as a risk-based number yet, and that's unfortunate. And I've been using Qpcr semi-online now for a year. It is incredibly informative. It tells you when your system becomes weaker in terms of protection. I think it's the future of control. I'm not sure how to bring it back to risk yet. And there are more people more knowledgeable about that may have a better answer.
Is Frédéric Hamas from Airbag online?
Hello, Frédéric.
I don't know if Frédéric is here. Are you online, Frédéric?
He will have his opinion on that. We've discussed.
Yeah, because it would be interesting to jump in the discussion, but we can do that in the round table afterward. Is there another question from a participant? Yes, we can go for the next one.
We have one question from Dr. Galliano. So how to make effective disinfection methods when Legionella is coupled with hosts eukaryotes, and are there any strategies?
I think the strategy in the NASM report, we had a lot of discussion. I was more of the engineering side, brought to a lot of infection control specialists from across the US and internationally, some Europeans. I think what we came out with is, at this point, we needed to provide guidance to how we can do it now. But the future of control in cooling towers and hot water system, it's not in controlling Legionella, it's in controlling hosts. Anything we can do to control hosts in the biofilm will control the risk. So when you ask me what can we do, we need to gather this incredible information that these new tools offer, so that we can show that this is the way forward. It's not the right way to measure Legionella by culture to bring a system under control. And it's not any better when you do qPCR. You're looking at the result of the problem. You're not tackling the cause of the amplification, which is the presence of hosts. So hosts are the future, but those methods are quite hard if you use the traditional methods. And but 18S has provided quite a bit of information. We need to move on to show how we can control hosts this way and provide a stable community that does not let NTMs and Legionella grow to high numbers.
Thank you very much, Michelle. Do you have the possibility to stay the whole session up to the end,
Yes.
or you are lecturing?
Yes, I'll mute myself, and I'm really happy
so
to
at the end of the session, we'll have a roundtable discussion, and I think that the point that you bring will lead to a live discussion. Thanks so much for your presentation again.
Thank you.
I let the word to Sophie now and Sergio, who will present the next speakers for the 10-minute presentation.
We will have the next speaker, Chen Dan, from Nanyang Technology University, will introduce us about the effects of extended stagnation on the quality of drinking water in a tropical above-ground network.
Good morning and good afternoon, everyone. Thanks for joining this presentation about the effect of extended stagnation on the quality of drinking water in an above-ground network in the tropics. In the past 2 years, the global pandemic caused by COVID-19 prompted work from home notices worldwide, which in turn result in closed offices, municipal buildings, or university campuses. These closures have been reported to alter the drinking water demand patterns and lead to widespread stagnation. Such stagnation situation creates.
Sergio, there is a problem with the online. We don't see anything about this presentation. So maybe what we can do is to tell all the participants to go in the program, and you click on the name of Dan Schenk, and there will be the video that is there, because the problem is that what that person uploaded, it is not uploaded in the right position, Nicole and Romy. Yeah, but it's not in the right. Can you go back in the program? There is no button watch pre-recording here. So let's solve this. Effect. Which one is it? Stagnation. Yeah. The problem, it is embedded, and we cannot put full screen here. It does not appear in the right. Maybe I suggest that we move to the next speaker and we do Dancheng in the end. And if you can solve this in the meantime, Nicole, this would be great to put in the website at the correct position. Thank you. So let's move to the next speaker.
So now we switch to the next speaker, Liu Xiaoxia from WESA's European Center of Excellence for Sustainable Water Technology. She will show us how the microbial calcium carbonate precipitation connects the biofilm and scaling process in drinking water distribution system.
Hi, welcome everyone to my presentation.
Can we show the main screen?
Well, it's the Senate cell who can do that.
Yeah.
We cannot do anything.
Hi, welcome everyone to my presentation. My name is Xiaoxia. I'm from WESA's, and today I want to talk about the microbial calcium carbonate precipitation process and how does it connect biofilm and scaling in drinking water context. So drinking water is actually a very special environment for bacteria because the organic nutrients is at a very low level, but the mineral concentration actually suits biology because the purpose it has to be suitable to for people to drink. And two common problems associated with drinking water distribution systems or biofilm scale, where biofilm cause more biological problems, such as like regrowth of pathogens and the threats to water safety. And the scale is more about the physiochemical problem, which is associated with pipe corrosion and the loss of waterhead and obstruction. But actually, if you have a look on the inner surface of a drinking water pipe, then you will realize that the bacterial cells and these mineral deposits are actually together location-wise. Then I start to think like whether it's possible that the bacteria can actually have a very strong interaction with these minerals in drinking, such as calcium content in water. And actually, during the literature review, I realized that the calcium carbonate should not spontaneously precipitate in drinking water system because they are most often in undersaturated condition. And of course, you always have the problem with the biofilm that it's difficult to remove and they're resistant to disinfectant. So here are two images under SAM. One is the calcite calcium carbonate scale, and the other one is bacteria biofilm. And it's very difficult to imagine that these two processes can have a connection somewhere. However, if you get a petri dish enriched with calcium nutrients and try to get a drop of drinking water there, then you will manage to isolate bacteria that can induce the formation of calcium carbonate crystallization. So here are the two species that are isolated. One is Fingobium in rod shape, and the other one is Micrococcus, which is in coccus shape. No precipitation happens spontaneously without the presence of bacteria. If you have a closer look on the development process time-wise, then you will observe that in the first 22 hours, the bacteria are very busy with growing by themselves, and you barely observe any interaction with calcium there. But from 48 hours, when they're relatively more established, you start to see this calcium concentrate on the surface of the biofilm. And if you wait for even longer, then you see a very thick biofilm, lots of crystal embedded in it, and the very big crystals can also be found on the surface of this biofilm. And if you wait longer, the crystal even grow bigger. And these crystals are very closely related to the bacteria cells. As you can see, you can find back their caves in their cell shape on the surface of the crystals, which here Stringobium is a rod shape and the Micrococcus is a round shape. We also observed that the transition of calcium carbonate crystal polymorph has been observed for this Stringobium species, where Viterite is the least stable form of calcium carbonate can be spotted, but also the calcite, which is the most stable form, and the transition state in between can also be spotted in the same sample, meaning that there's a transition of calcium carbonate phase happening in the bacterial medium solution. The same process have not been observed for Micrococcus. Only Viterite form of calcium carbonate has been observed, which indicate that maybe these two bacteria are doing different things. So when we look at the chemistry of the bacteria, we see that when the cell is growing, the ammonium is increasing and the pH is increasing because of ammonium, and calcium is getting precipitated. But the precipitation of calcium stops when the cell growth stops, even though the ammonium and pH is still increasing, which means that the amount of calcium that it gets precipitated is connected with the cell number instead of with the chemistry in the environment. For Micrococcus, not so much ammonium was produced and not the pH was increased, which really shows that a different mechanism is going on over here. And what is also interesting is that the calcium concentration only starts to decrease after the bacterial cell reach a certain density, even though the cell metabolism, such as producing ammonia, has been there for since the very beginning. So the chemistry that we have seen from those graphs are actually how the amino acids get oxidized into ammonia, carbon dioxide, and the water, where the ammonia can increase the pH and shift into ammonium, and that shift also the equilibrium of calcium of carbon dioxide into bicarbonate. And if you happen to have calcium in the environment, especially in the neighborhood, you will start to have calcium carbonate crystals form on the cell surface. And for the coccus, we observe a lot of polymers on the cell surface from sand. So we highly suspect that even though they do not produce a lot of ammonia, bioabsorption can be the main mechanism there. So the take-home message of the presentation is that the drinking water bacteria can induce and even control calcium carbonate precipitation process, and they even have different mechanisms for it. Thank you for all the colleagues that have supported me in this research, and let me know if you have any questions.
Thank you very much, Xiaoxia. And is there a question from somebody online, on site, in the chat? Are you online, Xiaoxia?
Hi, yes, I'm here.
Yeah, hi, thanks.
Can you hear me?
Pardon? No, this is Xiaoxia. Yeah. So then, I'm not a specialist of drinking water systems. So what is the problem with scaling in drinking water distribution systems? And how the approach that you have studied here can help solve this scaling? So what should be the solution then now to avoid scaling in drinking water distribution systems?
Hi, thank you very much for the question. Actually, the scaling problem for drinking water really depends on the calcium concentration of the local drinking water, which actually varies a lot at different places. And as Michelle already showed that for some drinking water system, there's also the temperature fluctuation, and the calcium carbonate actually the solubility decrease when the temperature goes up. So for a very long time, the scaling problem has always been seen as a problem from the increase of temperature. So it has never been associated with any microbial activities in the system. So in the experiment, the isolation that I did was actually from tap water, and later on we inspect some pipe surface as well of drinking water, and I could actually spot a lot of calcium carbonate, but also other iron, other minerals such as like manganese or iron, which also was interacting with bacteria. So solution wise, I'm actually my PhD was working on the application of magnetic field to solve this combined scaling and the biofilm formation problem, and so that is like the next step that we are preparing the manuscript to provide a solution for it. But I think the good understanding of the system and of the processes that is taking place in there is a good start for the solution hunting. Is that?
Thank you so much, Xiaoxia. Thanks again, and please stay online as Michel for the breakout discussion afterward. I suggest that we go back to Dan Sheng because now it is solved online. We can launch the presentation.
Yeah, thank you.
Thank you.
Zeb will share the video because we were having issues with ours. So no.
Yeah.
Good morning and good afternoon, everyone. Thanks for joining this presentation about the effect of extended stagnation on the quality of drinking water in an above-ground network in the tropics. In the past 2 years, the global pandemic caused by COVID-19 prompted work from home notices worldwide, which in turn result in closed offices, municipal buildings, or university campuses. These closures have been reported to alter the drinking water demand patterns and lead to widespread stagnation. Such stagnation situation creates ideal growth conditions for waterborne bacteria associated with the decay of the secondary disinfectant in the chloramine system. The influence that stagnation can have on microbial communities of built environment has been previously studied from household purifiers, plumbing systems for up to 5 to 6 days of stagnation, drinking water networks, urine network, flushing events, or fire hydrants by various groups. The impact of biofilm on the bulk water due to extended stagnation from national or regional knockdown remains unknown. The aim of this study is to understand how extended stagnation in the absence of monochroming influences potable water quality, to assess the changes in biofilm in biomass with assuming operations, and to determine if there is any quick indicator for water quality evaluation. To answer these three questions, an above-ground testbed was created early last year at Nanyang House NTU in Singapore. The whole testbed is isolated from the actual distribution network. There are 7 identical PVC pipe sections, outlets 1 to 6 for sampling and outlet 7 for maintenance. These outlets can be collected in parallel or in series. There is an additional sampling port outlet 0 near the end of shared pipe section. The source water for this testbed study is taken from one tap installed on NTU mains. Potable water in the tank is replenished continuously with little amount monochroming, about 0.3 to 0.8 milligram per liter. It travels from tank to the end of outlet 0 and then shared by all 7 replicate pipes. This drinking water network testbed remains stagnant from April to July 2020. 3 experiments were performed based on this testbed before stagnation under laminar flow condition, post stagnation under laminar flow condition, and then hard flushing to simulate reopening of the network. Flow cytometry, in short form FCM, with live dead staining was used to obtain the bacterial cell count within 24 hours. The fingerprint information based on FCM was also used to derive microbial diversity information with the help of the phenol flow algorithms in R. The following physical, chemical analysis was also performed immediately when the samples were collected. Bacterial cell count in the outflow water based on flow cytometry was used to measure the biomass concentration inside each pipe. The left y-axis in this slide represents the bacterial cell count of bulk water, and y-axis and the right y-axis the ratio of intact cell counts over total cell counts. Purple square represents the total bacterial cell count. Green circle represents the live bacterial cell count. Red triangle is the dead cell count, and blue diamond represents the ratio of intact cells. As we can see here, the 4 panels refer to the biomass condition of 4 different pipe steps, 1, 3, 4, and 6 under laminar flow conditions before extended stagnation. The log value of TCC range around 4.6 to 4.7, and the ratio of intact cells remained less than 20% during the 3-hour sampling time. In the post-stagnation, 2 tests were conducted. Test 1, stagnant bulk water samples were collected at time 0 and after 60 minutes, respectively, from the small tubing at flow rates of less than 0.1 liter per second in order not to disturb pipe biofilms. Test 2, the pipes were flushed separately at flow rates of approximately 2 liters per second to simulate the hydrological conditions associated with reopening of public buildings. About 250 ml of bulk water samples were collected in sterile Durant bottles in 10 seconds interval, up to 60 seconds, followed by sampling every 30 seconds for 5 minutes. More intensive sampling frequency was implemented to the last pipe outlet 6. The middle and the right plots show the profile of stagnant bulk water measured twice by FCM. It also suggests that there was no significant difference in the measurements, indicating no biofilm detached at this flow region. Again, purple is all cells, green is live, red is dead, blue is ratio. The cells in the water were mostly alive, giving the high ratio of around 40 to 70%, and the total cell count in its log form has increased to 5.3 to 5.4, and there's about 0.7 increment than before stagnation, equivalent to about 5 times higher than before. These plots shows that conductivity decreased over time during the hard flushing event for all the outlets except for outlet 2. This is likely caused by the higher flow rate of outlet 2, shown in blue square, which was tested first, and it was about 5 times higher than all other outlets. The 6 plots in this slide shows the profile of bacterial cell count during the 5-minute flushing window of all 6 replicate pipes. As we can see, the highest bacterial cell occurred during the first minute and then gradually stabilize. Not only conductivity decreased over time during the flushing event, so did the second order Hill number, in particular during the first 60 seconds. This parameter is commonly used to describe the bacterial diversity and takes account the abundance information. In this study, we obtained phenotypic diversity from the flow saturation data according to flow algorithm in R. The cluster of bulk water samples based on respective phenotypic beta diversity was visualized using principal coordinate analysis. The left panel shows the cluster of all samples, and the right shows only the flushing events. It showed significant difference for the bacterial community before and after extended stagnation, as well as microbial community, all outlets in the first 60 seconds, or called initial flushing, and 61 to 600 seconds, or called post-flushing. The bulk water community shift clearly before and after stagnation. After flushing, the community seemed to be closer to the events before extended stagnation. As we saw earlier, both total cell count and conductivity showed similar decreasing trend with time. Here, a regression model was applied to these three parameters to see their relationship, and the plot implies a polynomial relationship between total cell count and conductivity. This graph shows very clearly that at lower conductivity, there's no relationship, but higher conductivity indicates an increase in total cell counts in the water. The higher conductivity was only seen in the first 60 seconds, as indicated by the color coding. Each 1 unit of microsiemens per centimeter increment in conductivity implies a 2.4% increase in the total cell count in its absolute number, controlling for time. To conclude, we managed to capture the detachment of biofilm occurred in the early stage upon restarting of operation using flow cytometry. The quantification results from FCM shows massive detachment of biofilm in the first 60 seconds. Thus, the first minute of water upon restarting of operation should not be used for drinking purpose. The polynomial relationship between conductivity and total cell count indicates that conductivity can act as a quick indicator for microbial bulk water quality for operators. The detachment of biofilm could be measured using FCM phenol flow-based approach. Bulk water samples taken during the flushing experiment provide additional evidence that the reopening of public buildings after extended stagnation can lead to the potential exposure of consumables to higher numbers of total and variable bacteria, as well as a significantly different microbiome during the first minute of opening. Thank all help and comments from all members of Cluster 1 and our intern students. Special thanks goes to my former PI and collaborator, Prof. Andrew Witter from MIT, and my dearest supervisor, Prof. Stephen Woods, for their detailed guidance on the design of the experiment. We'd like to thank all kinds of help and discussion given by CLC colleagues from time to time. We'd like to acknowledge Prof. Icobun and his team for their help in demonstrating the phenol flow approach during our visit in Ghent University. Thanks to the CLC core funding and JWAF's seed grant for MIT. Last but not the least, thank my colleagues, Dr. Matt Stanfield, for all your criticism plus suggestions and support. I welcome any question or comment if you have.
Congratulations, Dan. It's a very nice setup of contributors that you have here. It should be really fascinating to work with all these different persons. Are you online, Dan?
Yes.
Yes?
I am. Yes.
Is there a question somewhere in the chat, the Q&A, drinking water specialist in the room? So, with all these different questions about these biofilms and the microbial ecology, we can come back to the roundtable afterward, but what is the problem in the end? Is it the presence of the biofilm or the functionalities of the populations that are present in the biofilm? So what do we want to fight? Is it to not have a biofilm, or to not have a biofilm of certain types of populations with certain functionalities that we don't want?
Sorry, can you repeat your question?
So in your stagnation experiment, so at certain point, you showed some pictures with biofilm inside the pipes. And with the different talks that we had before, we saw scaling microbial ecology endpoints to address. But is it any biofilm that we want to get rid of, or this is a biofilm with specific unwanted organism that we want to get rid of? And is there a way, based on your experiment, to think further beyond the biofilm itself? What is a good biofilm? What is a bad biofilm in these drinking water stagnant systems?
I think this is a very interesting question. For the biofilm, as we all understand, biofilm is present at many places and along the whole drinking water distribution networks. So the aim of this study is to tackle what happened inside this inside the drinking water networks after extended stagnation. So our focus is not to remove, not to find out what is the good biofilm, what is a bad biofilm. The goal of this study is mainly to answer how we can measure the biofilm detachment and what will be happening inside the network during this extended stagnation period, and what customer will expect to receive.
So based on your findings, what would be your suggestion for the drinking water practitioners? How can they manage this detachment? It's by managing the flow of drinking water in the pipe. So what should they do? Can you derive some hint for practices?
Yeah. So one of the takeaway message from this study is that we can use conductivity as a quick indicator to measure the cell count, to estimate the total cell count.
And thank you. And we'll come back to this question with the different contributors as well afterward. Thank you so much, Dan. And we move to the next speaker, Sophie.
We have the next speaker, Marcus Freud from Sweden Water Research, and will show us how to use online flow cytometry to support safe and smart maintenance of drinking water pipes at full scale.
Is it pre-recording?
Yes, Seb will share the pre-recording video.
Seb who shares?
Yeah.
Hello everyone, my name is Marcus Freud, and I work as a research leader at Sweden Water Research in Lund, Sweden. Sweden Water Research is an R&D company owned by 3 water utilities in southwest Sweden. Today, I'm going to speak a bit about the project I'm involved in, supporting safe and smart maintenance of drinking water pipes at full scale with online flow cytometry. This project is a collaboration between the water utilities Sveaseed, Sydvatten, and the R&D company Sweden Water Research, as well as Lund University. The project is supported by funding from Svenskt Vattenutveckling. Before we dig into the details, I would like to frame our work a little bit. Firstly, by new pipe installations, I mean all work where new pipe sections are installed, such as repairs, replacements, and new pipe sections in newly developed areas. These pipe installations are a potential source for contamination. Therefore, before a new pipe is connected to the drinking water distribution system and our customers, the water in the pipes must be quality tested. The current methods of maintaining safety in new pipe sections involves doing a barrage of standard analyses. Currently, the time-limiting step in the quality control are heterotrophic plate counts, or HPCs. Swedish water utilities rely on HPC methods according to international standards to assess overall bacterial growth and growth potential in drinking water. That is, they're using 2 different HPC protocols where results take 3 or 7 days to get. Further, the standard practice is to wait for consecutive samples to be approved on 2 consecutive days for the pipe to be allowed to be connected to the distribution system, leading to a minimum of 4 to 8 days before the pipe installation can be approved. During the period in which HPC results are awaited, the pipe is continuously flushed with drinking water. This water, unfortunately, goes to waste. One of our project goals is to be able to do our quality analysis of pipe installations faster. How can we do that? Well, there are new techniques available to us that could be used to speed up the quality measuring process. These methods can either directly yield faster results or increase our understanding how the microbiology of new pipe installation works, so that we can improve our routine protocols and speed up the process. In order for us to maximize this potential, we need to support the discussion and development of new analytical methods and practices in the industry in a wider context. Why is doing faster quality analysis of new pipe installations important? Well, there are many potential benefits from shortening the waiting period before connecting new pipes to the network, and I will briefly mention a few. Faster return to service for our customers, for instance, when we do repairs or replacements. Saving water from being flushed, which has economic, environmental, as well as public goodwill implications. Sometimes, in certain regions or in times of water shortage, maintaining flushing for extended periods of time can be difficult or even impossible. And this is expected to be a more common problem in the future as our climate changes. The societal impacts, especially considering accessibility in urban environments, can be very large. For instance, when pipe replacements lead to diverted traffic and closed roads. So today I want to talk about a case study from the part of our project that concerns new methods, specifically flow cytometry or FCM as a method of assessing water quality in new pipe sections. We chose FCM as a first method to test, and as we had experts available in our respective organizations, we already had some instruments available to us, and it's a fairly well-tested method internationally. It's also a method that gives us results in approximately 15 minutes, compared to the 3 to 7 days for the currently used HPC methods. It is, however, important to note that there are other methods of potential interest available on the market, and they should be evaluated moving forward. Unfortunately, I do not have the time to go into all of our case studies, so I will focus on just one. In the so-called Tecumotop leak, a leak occurred on a large transfer pipe from one of the larger waterworks in the region, and it required replacing a section of pipe. As seen in the schematic on the left, 2 locations, A and D, were up and downstream of the leak, and portable automatic flow cytometers were placed at these sites after the repairs had been completed and the flushing of the pipe had begun. HPC analysis was done on batch samples from A and D, as well as from sites B and C, which were closer to the repaired section. So what were the results from this case study? In the figure on the left, the flow cytometry data from the downstream point D is shown in green, and the upstream point A is shown in orange. As you can see, there is a period shortly after starting to flush the pipe where the bacterial load is unstable and on average significantly higher than at the upstream sampling point. However, this returns to level just slightly above the upstream sampling point after approximately 2 days and remains stable for the duration of the flushing period. The inlay shows the same data, but without the initial sampling points shown, just to highlight that there are in fact variations in this data as well. Interestingly, the drastic increase in cell counts is not clearly visible when looking at the HPC data shown on the right. The upstream reference sample, in orange, is very stable and near 0 colony-forming units per milliliter during the entire period. However, some of the downstream sampling points, including just upstream of the leak, has an increase in HPC counts after approximately 9 days, something that is not clearly visible in the FCM data. It is quite interesting to ponder what these variations in HPC counts really mean, especially given the context and what they actually tell us about the water quality. That something happens after the repaired section seems to be clear from the FCM data. But can the data be used further? I won't go into the details of this image, but the link here leads to Monsamore's master thesis report, where the details are explained. So very briefly, on the left, you see typical raw data outputs from the FCM. This data yields a fingerprint, and then each fingerprint can be compared to one another to get a measurement of their differences, which is shown in the Bray-Curtis dissimilarity plot on the right. Each point represents the profile of a given measurement, and the closer the value is to 0, the more similar the fingerprints are. The upstream, shown in orange, and downstream profiles, shown in green, do not overlap, which indicates that there are in fact distinct bacterial communities. Within the tested timeframe, the bacterial communities before and after the new pipe section are not homogenized. Further, the earlier sample points after the leak are drastically different, as we could see in the cell counts as well. One interesting detail is that around day 7, there is a short-lived change in the bacterial composition in the downstream sampling point, something that did not appear in the HPC analysis and was not readily visible in the total FCM cell counts either. This was a small sample of the questions we're currently working on in this project, with more case studies and testing on different types of new pipe installations underway. So to conclude what I've presented today, FCM can give us more information and more detailed information, but it is complex and requires more knowledge and processing to use effectively. The FCM methods reveal that the HPC counts in new pipe installations are not necessarily indicative of changes in overall bacterial load or drastic differences in bacterial communities. So can new methods, for instance FCM, give us better information and help us complete pipe installation works faster? This is still an open question, and we will continue to investigate this during the course of the project. With that, I would like to thank you all for your time. Please visit our webpage for more information about this project and our other projects. You can also contact me in case you would like to know more about this project or if you have any questions. Thank you.
Congratulations, Marcus. Are you online as well?
Yes, I'm here. I hope you can hear me.
There is a question, I think, from Michel Prévost. Sophie, you can ask. Oh, it was from the previous. Okay. Yeah.
David, oh là là.
Make a TV show. And, yes, so I know, for instance, from Switzerland, we spoke about Frederick Hammes before, that the flow cytometry method is validated by the Food Administration, let's say, and Water Administration in Switzerland. How is it in Sweden? Is it a standardized method, FCM, or is it the first time that you implement it in your projects?
So unfortunately, the Swedish Food Agency do not allow FCM as a viable method to assess either risk or even cell count. So we have to completely rely on HPC methods for our analyses when it comes to taking new samples. And that's the core of the problem. So part of this project is obviously to influence our agencies in the right direction. We need new technologies. We need faster answers, simply put. And this is one step in that direction. We are unfortunately not as far with FCM as Switzerland is, I'm afraid. So it's not accredited in Sweden, no.
So what is needed to move forward with the authorities?
Well, that's a very difficult question. I think the authorities will obviously need a very good scientific basis provided by the water utility community in Sweden to allow us to use this data in a way that we would like to use it to obtain more information, simply put. But whether or not we can reach that goal in the foreseeable future remains to be seen. It's also regulated on an EU level and so European Union level, and of course the local Swedish authorities will also have rules and regulations that must be adhered to at all times. So it's a difficult process for the utilities to really move this field forward, I think.
And if I would be an early stage PhD candidate aiming to sample a drinking water network, what should be the sampling frequency that I should involve to survey over a year drinking water network and the dynamics of microbial populations there, and to detect potential issues?
That's a very good question, and one we're constantly tackling ourselves. I think finishing a PhD doesn't really help you or give you the answers in terms of your sampling strategy here, because you have seasonal variations, you have daily variations, especially in small suburban areas where the water usage varies greatly over the day. You can have drastically different effects on your flushing rates and your flow rates in the network. So I think it's very different from small rural areas to big urban areas, times a day, and of course seasonal. So it's short answer is I don't know.
Thank you, Marcus, and we may come back to that during the breakout. Let's move to the next speaker. Is it on-site or online? Online.
So, RJ, we have the next speaker, RJ Bosman from Radboud University, will introduce the effects of methane on nitrogen removal and microbial composition of rapid sand filters. So,
Yes.
if you are online, the screen is yours.
Let's see. Let me share my screen. See if this all works. Can you guys see my screen?
Yes.
Great.
Because it's not yet in YouTube. Now it
okay.
is.
Yeah.
You can start. Thank you.
Great. So good afternoon, everyone. My name is Ali Boersma. I'm a PhD student at the Department of Microbiology of Radboud University in Nijmegen. And I will present to you our research on the effect of methane on nitrogen removal and microbial composition of rapid sand filters used for drinking water production. So let's start out with a very brief introduction on rapid sand filtration. Rapid sand filters basically are a layer of sand where water, which most of the time is aerated, is passed through. And then a mixture of chemical and biological processes take place to remove contaminants and produce either clean drinking water or water that is ready for downstream treatment. The main contaminants that are removed are any methane that is left after aeration, iron, ammonium, and manganese. So there are two main types of rapid sand filters. Firstly, the submerged rapid sand filters, which have a permanent layer of supernatant on top of the sand bed, and then the trickling rapid sand filters, which are also called dry filters, which don't have a permanent layer of supernatant on top of the bed, and the water is trickled on there. Most of the time, these trickling filters are used when the ammonium load of the water is so high that even water that is fully saturated in terms of oxygen doesn't have enough oxygen to oxidize all the ammonium. And this trickling setup allows for more contact with oxygen. So the drinking water treatment plant we investigated has multiple approaches to producing their drinking water. They have several treatment lines that look like this, where there's water which is highly aerated, so it's very low in methane, and it passes through two submerged rapid sand filters. And they have one line that looks like this, where water is not aerated, so it's very high in methane concentrations. And the first filter is the trickling filter, and it then passes to a subsequent submerged rapid sand filter. What the operator saw was that nitrification was performing well in the submerged rapid sand filter, but was not performing at all in the trickling filter. And this gave us the idea that this might be a nice system to study what the influence of methane is on nitrification in a full-scale system. So what we did, we sampled in two of the lines that contain two submerged rapid sand filters, the first rapid sand filter. So not the first and the second, but two times in two lines the first rapid sand filter. We sampled here the top, the middle, and the bottom. And for the line with the trickling filter, we sampled both the trickling filter and the subsequent submerged rapid sand filter. We took water samples, which we used for ammonium nitrite and nitrate measurements. And we took sand samples, for which we measured the iron and manganese coating. We performed activity assays, and we did metagenomics. For this presentation, I will focus on the ammonium measurements, the activity assays, and the metagenomics. So for the ammonia measurements, these are measurements done on the full-scale system. First, you see the raw water, where the ammonium concentration is about 0.75 milligrams per liter. Then in green here, you see the ammonium concentrations after the submerged rapid sand filter step. You can see that the ammonium is largely removed. However, after the trickling filter in pink, you see that the ammonium concentration is still quite high, more than 0.5. And then in both the after filters, so both after the submerged filter and both after the trickling filter, you see that ammonium is completely removed. Then we performed activity assays. So we have the submerged filter 1 and 2, the trickling filter, and this is the submerged filter that comes after the trickling filter, which were incubations spiked with ammonium, where we then measured the ammonium conversion rate for the top, the middle, and the bottom sample point. And you can see that the two submerged filters both show conversion of ammonium and are very similar to each other. The trickling filter, which receives water that is very high in methane, shows very little ammonium conversion. However, the submerged filter after the trickling filter shows at the top the highest ammonium conversion that we see in all the incubations we did. We then did more activity assays, but this time we did not only feed ammonium, but we also fed ammonium and methane both. So in brown it's just ammonium, then in gray ammonium and methane. And you can see that in every filter where there is ammonium consumption, when we add only ammonium, when we then add methane, the ammonium consumption lowers quite drastically. So it seems that also in our incubations, the presence of methane is hampering ammonium oxidation. Then for the metagenomics, what we did was a gene mining for the RPUB gene, which is a very good phylogenetic marker, and for both the functional genes for the first step of ammonia oxidation and methane oxidation, so the AMOA and the PMOA. And we then performed read mapping as a way to quantify how much those genes were present. So in this figure, you see the RPUB abundance for the submerged to submerged filters, the trickling filter, and then the submerged filter that comes after the trickling filter. And we group the RPUBs based on their predicted metabolism. In red, iron oxidation, in green, methane oxidation, and in blue, nitrifying bacteria. When you look at the two submerged rapid sand filters, you see that there are nitrifying bacteria present, a small amount of methane oxidizing bacteria, and there are also iron oxidizing bacteria present. And the biggest difference you see here in the trickling filter, where you see a lot of methane oxidizers present, and a little bit of iron oxidizers, and almost no nitrifying bacteria. However, then in the filter that comes after the trickling filter, you see a very large amount of nitrifying bacteria, especially in the top. And this is also where during our incubations we saw the highest ammonia conversion rate. Here we looked at the ammonia monooxygenase abundances, and we grouped this whether they were the AMOA was associated with canonical ammonia oxidation, so just ammonia oxidation to nitrite, or with COMAMOX, so full ammonia oxidation to nitrite and then nitrate. And in the submerged, the submerged rapid sand filters, you see that both the classical two-step ammonia oxidation and COMAMOX is taking place. However, in the submerged filter that comes after the trickling filter, you see that it is almost only a two-step nitrification that is taking place there. So COMAMOX is more prevalent in the submerged filters that is not in the trickling filter line than in the submerged filter after the trickling filter. What I would like you to take from this is that we show that the presence of methane inhibits nitrification during rapid sand filtration, for which we have strong indications in the full-scale system, and we show in our smaller incubations, and that the presence of methane has a large influence on the microbial community. We see more methane oxidizers, less nitrifiers, and also less iron oxidizers there. And that in the purification line, for the trickling filter line, we see more two-step nitrification, and in the line with the submerged filters, we see more COMAMOX. I would like to thank these institutions and these people for their all their help, and in special Francesc Corbera Rubio, which is my main collaborator on this project. So, and also thank you all for your attention, and I'd be happy to answer any questions.
Congratulations, Ali. Thank you. I have a rapid question. Could you have anticipated, based on wastewater knowledge, that you would not have any nitrification when you don't have a lot of air in the water and when the organic matter is high in the water? Because from wastewater treatment, we always say if we make respirometry test, if there is a lot of organics, there is no chance for a nitrifier to grow. So it means that the oxygen is scavenged by the ordinary heterotrophic organisms first. So could you have already predicted that based on these types of knowledge from respirometry tests?
That's a good question. I don't know. The water that comes in is not high in organics, so the groundwater. But of course, there are organics coming off in the biofilms that is on the filter medium. I think a larger role is the competition for methane that is there, and the competition for oxygen also that the methane oxidizers will present to the nitrifiers. But the organics, I don't know exactly what their role is. Yeah.
Yeah, I would think until there is still methane in the system, organisms that can oxidize methane will use the oxygen, and nitrifiers have no chance to grow because they are slower growing.
Yeah, so we think that this is indeed part of it. There's also competition maybe for copper, which is a cofactor both for the AMOA and the PMOA. So yeah, could be, or a combination of all those things. Yeah.
Okay. Thank you, Alier. And let's move to our last speaker, Francesc, that you named just before. Floor is yours. Francesc, it's an on-site presentation. Where do you present it at the console? Can you help Francesc getting set up?
Yeah, if you have this on, then you have the camera. Is this one?
This is the presentation. Yes. No, not that. This.
This one is working.
From the Senate Salle, is it okay if Francesc used the portable microphone or not? But switch off this one then. Yeah.
Does it work? Yeah, it works. Great. Hello. Hello, everyone.
No, wait. Senate Salle, we don't see the presentation of Francesc online.
Yes, we do.
We see now? That's fine. No, we see it on YouTube, but I don't see it in the session block.
You have to click in Senate Salle.
And maybe explain to the participants, because I'm like them.
So you have to go to the profile of Senate Salle and click. So then you have the presentation and the camera.
Okay. So everybody should click on the Senate Salle profile on the Jitsi to see the slides. And it's also live stream on YouTube. So please, floor is yours, Francesc.
But can people see me?
Yes.
Yeah. Okay. So hello, everyone. One of the conclusions of Dan Cheng, who presented just before, was that during the first 60 seconds of flashing a pipe, all your biofilm detaches. And I feel this is also what happens with the audience when you present. You know, during the first 60 seconds, most of the people will stop listening to you. That's why I wear a yellow shirt, so you know it catches your attention. I also wear it because I work with Mark, and he has taught me many things, and I'm still learning a lot. But one of the main ones is that you have to wear vivid colors when you present. You know, he's very famous for the red blaze. So with this introduction, let's start with the presentation, in which I'm going to talk about groundwater, anaerobic groundwater specifically, because it is the main source for drinking water. There's no pointer. Oh. Yep. Okay, I'm ready. Which is the main source of for drinking water production. And it is the main source because it has two great advantages. The first one is that it has almost no pathogenic bacteria, and the second one is because it has very stable temperature and concentration of contaminants. And these main contaminants are the reduced forms of nitrogen, which is ammonia, manganese, and iron. And if we want to remove these contaminants, we have to oxidize them, because once you oxidize them, they precipitate, and then they are trapped in a filter. So the first step to remove them is to oxygenate the water, because it's fully anaerobic. If you want to oxidize, you need oxygen. So you pass the water through a plate aeration, which is a platform that looks like this. The water flows from the top to the bottom, and air flows from the bottom to the top. So you reach saturation levels of oxygen. And then once the water is fully aerated, you send it to a Dryfta filter, which is a huge tank full of sand grains. And then in the sand grains, and with the oxygen, your contaminants are oxidized, they precipitate, and they're trapped within the filter, which yields clean drinking water. The problem is that all of this, which I explain very quickly, happens via many different reactions which also interact with each other. So you basically, when you try to understand how the Dryfta filter works, you have a myriad of different reactions which are difficult to understand. So what I wanted to see is, where do the reactions happen in the Dryfta filter? And how do they happen? Which microbial communities catalyze these reactions? Which is what I try to do. So quantitatively assess the removal of iron, manganese, and ammonia, and which microbial communities drive this removal. And I did it in two different process schemes. In one field, in one plant that uses only one single sand filter, and another that uses two sand filters with almost identical concentrations in the influent water. In the first case, the first filter looks like this. It has two types of sand in the same bed, which have different colors. And this color is given by the coating. And the coating is given by the contaminants that have been reduced. When you remove iron, you get a orangey reddish color, which is mainly at the top, as you can see here. And when you remove manganese, you have a blackish color, which is what you see at the bottom. And then we have a two sequential filter system, which started with identical sand. But after some years of operation, when we opened the filters, we saw this. The first one they look very different. The first one has this orangey color, and the second one has this blackish color. So as you can already see, that even though the processes are different, the process configuration is different, the sand looks the same. Right? You have two different compartments in both cases. So to understand where the removal of each component happens, we took samples, solid and water samples along the height of the filters. 3 in most of the cases, in this case only 2 because it was not possible to sample deeper. And let's see where the contaminants are removed. In the first case, we see iron, and in both cases, iron is removed in the first section of both cases. So after the orange coating, there was no iron anymore. So complete iron oxidation in the first section of the filter. Once iron oxidation was completely finished, manganese was removed. In the first case, in the middle, so in the black section, in the second case, in the second filter. And last, ammonia oxidation happened partially. Partially is a bit too much. A little bit of ammonia was removed in the first section, but most of it happened also after complete iron oxidation. So now I'm going to focus on, so we see again the compartmentalization. In the first section of the filters, iron is removed. In the second section of the filters, manganese and ammonia are removed, even though the filters are theoretically completely different. To follow a bit more where exactly the process happens, I'm going to focus now on the removal of ammonia. What we did is we took the sand samples at different heights, and we incubated them in batches with ammonia to see which was the ammonia removal capacity at each height of the filter. And this is what I'm going to show right now. We see that, again, the same compartmentalization. In the 2 first sections, we see very little ammonia removal in line with the concentration plots I showed before. And in the second sections, we see very high ammonia removal. What's also important here to see is that especially in the second section, in the second filter, the removal of ammonia is very stratified. We have the highest ammonia removal capacity at the top and the lowest at the bottom. So we see compartmentalization and stratification of ammonia removal. The next step in this case was to see, okay, we see ammonia is, the capacity to remove ammonia is stratified. Can we predict this with the relative abundance of the microbial communities. So to answer these questions, we sampled, we extracted DNA, we did metagenomics. And I'm going to show now a graph of the phyla results. There's no time to go through all the graphs, so let's focus. Follow me a little bit, follow the light. Even though the removal of ammonia was strongly stratified, the populations in the filter look fairly homogeneous. We didn't understand this at the very beginning, but now we think we have an answer, and the answer is backwash. So these filters are regularly backwashed very thoroughly to clean the iron that has been precipitated. What we believe happens is that when the filter is backwashed, the sun grains, together with the biofilm that grows on top of it, are homogeneously distributed through the filter. Maybe not every time you backwash, but this happens many, many times during many, many years. And the result is homogeneous distribution of the microbial populations. But then we still needed a method. But then we still needed a method, spoiler. We still needed a method to track activity. So then instead of tracking potential reactions, which is what you do with metagenomics or with any method that tackles DNA, we chose metaproteomics because proteins are the actual biological catalyzers of reactions. And this is what we saw. This is a graph of normalized spectra counts, heat map, and it can basically be read as relative protein abundance. Here on the x-axis, we have different genes that belong to the nitrification and denitrification pathways, but we're going to focus only on the very last one. You can read whatever you want, but I'm going to focus on the last one. In gray, we see the proteins that were only present in the metagenome, but not in the metaproteome. And here we see on the y-axis in a scale of reds, darker red means higher protein abundance. And then what we saw is that when we detected low ammonia removal capacity, we saw low abundance of the proteins that belong to the nitrogen metabolism. And when we saw high and stratified ammonia removal capacity, we saw high and stratified abundance of the proteins that belong to nitrogen metabolism. So metaproteomics, in this case, seems to be a good method to track activity. The take-home messages, the first one, there's clearly a shared biological and physical chemical stratification patterns in Rapitan filters, regardless of whether one or two Rapitan filters are used. And second, activity correlates with metaproteomics, but metagenomics does not, at least in this case. And we believe this is likely due to backwash. And since this is the last presentation of the session, I thought I could start with the panel with the discussion panel with a question, which is, based on this and your knowledge, is backwash beneficial or not for microbial conversions in Rapitan filters? I would love to hear your opinion on this. And last, and most important, I did this work with the help of my three supervisors, Doris, Michele, and Mark. And the project is part of the Sun Filtration Program, which is an initiative of NWO, which is the Netherlands Organization of Sciences, and Dunea and Vitens, two of the biggest drinking water companies in the Netherlands. Thank you very much.
Congratulations, Frances, for this excellent talk. And I should say that after all the technical issues that we had, it's very nice to see your talk. Yeah, it's really great. And a nice shirt as well. Please stay here. We have some questions for you. And you will participate as well to the roadmap discussion. So I suggest that we make the closure of this day with this session. So I convey maybe all the speakers already switch on your camera. And is there a direct question to Francesc from the room, from online, and then we move to the other speakers. And Niti, the young water professional, you can also join this session. We will make the closing directly during this session.
Nina.
Yeah, you need to come to the microphone. Yeah, there is a question from Nina from TU Delft.
Thank you, Francesc, for your very nice presentation. So you think that backwash is the problem, or at least why you don't see the stratification in the DNA. But how often does this backwash happen then? Because I guess it needs to be slower than the protein turnover.
Hello. Yeah, it depends very much on the plant and mainly on the concentration of iron. So the clogging is partially because of the iron that's precipitated and also partially because of the biofilm that grows due to the conversion of mainly ammonia. But iron is clearly the main trigger or the main reason why backwash is needed. If the plant has high concentration of iron, up to 30 milligrams per liter in the Netherlands, then you need to backwash every day or every couple of days. But if the concentrations are low, which was the case of this one, I think it's every week or something like this.
Okay, that's pretty quick. That's quite often, but you still see quite a clear difference in the protein expression then.
Yes. Yes.
Okay, thanks.
I have a question on the sampling. How do you physically sample the system? So they are so big, and we saw you on a tank with your lab jacket. So where are the sampling points, and what do you sample? Do you sample only the water phase, or you also sample the biofilm around the particles of the sand?
All the data I showed corresponds to the biofilm that's on the grains. So we had to sample grains. So to sample, basically, you need an army of people and a pit layer sample, which is like a huge device which ends with a cone. And then you, from the top of the filter, you push it all the way down. And you, with a hammer, you have to, And then it has a mechanism so that when you can open and close the cone that's at the bottom. So it's rather easy, let's say, to locate the position in the height of the filter in which you sample your sangulates.
How many sampling points can you take physically at the same time? Because I would assume that it can influence the flow regime in your sand filter. Or is there an impact of this carottage of the system?
So when I went here, I thought that 3 were already quite a lot of sampling points. Important, you have to do this after or before operation. So you cannot sample while the water is running, of course. But then Alje, who just presented, took like 25 sampling points from one single filter. So I think that the number of samples you can take really depends on your goal.
Thanks, Francesc. And I think you made a good connection to Alje when we can ask the next question to Alje. So you mentioned that you collaborate together, actually, and then there will be a question afterward from David. And so what do you gain from each other's collaboration? So what do you gain from Frances's side and what Francesc gains from Alje's side? So how do you complement each other in your science practice?
Yeah, that's a nice question. Of course, it's very nice to have just a different point of view and have a person to discuss with who's in the same boat as you are, started around the same time on a similar subject. And then again, of course, the labs where we work are specialized on different things. So what we're doing on this project that I just presented, where I presented the metagenomic data, I will give this data to Francesc and they will use it to do metaproteomics. So that's a very nice way to complement this data and not only see what they might be doing, but also what they're actually doing. So that's, I think, very cool. Yeah.
This brings the point about research collaborations and yeah, for these big systems that are very complex, I think it's the way to go. You cannot replicate too much this system either, but maybe there are other strategies. There is a question on site from David Calderon. To which speaker do you have a question?
To Francesc. Well, maybe for both, because you are working with omics in general. So I heard you said that mainly one of the reasons could be backwash. And then I was also wondering how easy it is to obtain DNA from these kind of samples. and how easy it is to obtain biomass from these samples. Then, how do you translate this into how reliable the results are from metaproteomics and from metagenomics? Because you were saying that you could see the effect better with metaproteomics and less good, let's say, from metagenomics. So how the extraction of whatever biomolecule you're analyzing is affecting this analysis?
So Ali, do you go or do I go?
No, go ahead, Francesc. You can start if I've got something to add.
Okay. Any kind of extraction from the sang range is, I would dare to say, extremely complex, because the sang grains are covered, as I said, with manganese and iron oxides, which are very much liked by DNA. So the DNA usually is easily adsorbs on these structures, and then it's very complex to get DNA. So you need to thoroughly optimize your extraction methods to obtain enough DNA and enough proteins. Ali is way better than me at doing this. He has always way higher titers, DNA titers than I have. But indeed, it could be something that biases our data. We cannot discard it, but we cannot go farther than what we have done. So we said, okay, let's try with both methods and see. Please.
No, which would mean that maybe for this kind of settings, you could say, or you could recommend for the future scientists working on this, use metaproteomics.
Yeah, I don't dare to do that. There's very good work on metagenomics on this type of systems, and I repeat, it depends very much on how much iron you have. There's a couple of excellent works recently, one from Alejandro Palomo from DTU, and one from Liana Pogoshan from Radboud University, in which they use metagenomics to study the community in the rapid filters, and they managed to get very good data. If I recall properly, they had rather low iron concentrations. So I think this helps.
Thank you very much.
Thank you, David, and thank you, Francesc and Ali. So let's move back to the other speakers as well. So here with Ali and Francesc, we have really seen about functionalities in terms of metabolisms that happen in a sand filter, for instance, nitrification, Comamox, or other types of microbial processes. So they are metabolizing, they are catabolizing the nitrogen or organic matter. And then with Michel and Marcus, we have mainly seen about methods and microbial ecology principles that we can apply to drinking water systems. And we've done on Xiaoxia, it's more about biofilm and scaling. How can we manage these processes in the tubing? So this brings me to this basic question that I have, and since many MIWI conferences, what are the functionalities that we need to tackle in drinking water? So from a wastewater scientist that I am more, we look at the catabolism, so nitrogen removal, organic matter removal, phosphorus removal, etc., etc. So these are main metabolisms that are exacerbated by the organisms, like what Ali and Francesc do. But in terms of Legionella, in terms of biofilm formation and scaling, what are the functionalities that you can get from the populations that are populating these biofilms? Maybe Michel, based on all these discussions, so maybe these 3 sets of presentation, what is your thoughts about that? How can we tackle functionalities in drinking water? Are you still here, Michel?
Yes, I am, but my microphone was turned off. Sorry.
Okay. And did you hear the question?
Nope, I did not.
Okay, so I will rephrase shortly. So in these presentations, we have seen, for instance, with Ali and Francesc, functionalities about nitrogen removal or carbon removal. We've done on Xiaoxia about biofilm and scaling. With you and Marcus about microbial ecology principles and methods that we can implement in complex drinking water systems. But this brings me back to a very old question that I have about the functionalities in drinking water. So what are the key targeted functionalities that we have to analyze in drinking water? In wastewater, it's quite obvious, it's carbon, nitrogen, phosphorus removal. What are the functionalities that we can seek from a genome of organisms from drinking water?
Well, it's a really good question, and I'll piggy up on one of the last questions. One of the things we thought was most exciting about our chlorination of cyanobacteria project is we wanted to show that low dosage oxidants, chlorine, peroxide, whatever, actually initiated a response by toxin production. It had been seen in one lab, and we thought, oh, this is so exciting. We're going to show this turn on and this ability through proteomics. So we thought this was great. The trouble is that when you're working with some of the drinking water samples, whether they're oxidizing cyanobacteria, as the example I just gave, or working with suspended biomass and distribution system, is your ability to extract DNA and extract RNA are really limited. And we hardly got 10% of our extracts that were good enough to give us a strong functional profile. And often in a series, when you impose some kind of stress, then you lost the signal. So the most important point that you wanted to get, you can't get. So to me, that's still in drinking water, it still is a challenge to answer you which function you would want to get rid of does not answer either the fact that which function would be a predictor of which risk associated, for example, with the growth situation of a given pathogen. So I don't think we have that answer right now. Perhaps if you're looking into preventing nitrification, then you could probably pin that one down, but I don't think in terms of risk reduction that we're there yet. And as I said, we're limited to our ability to really gather enough DNA to get the job done.
Yeah, and when you speak about risk reduction, I think it links well to Marcus when we discuss about how to convince authorities that we have to embrace some methods as well. We have the same issue with antimicrobial resistance. David, who asked the question before, will speak on Wednesday, and we'll speak about risk assessment as well. But how far are we to be able to address a risk, a microbial risk? Or how accurate are the methods? What are the methods that are useful to address the risk? In chemistry, we generally speak about those responses.
Yeah.
How do we apply this risk assessment in the microbial world? And how far are we to convince authorities that we can do a risk assessment and we can then mitigate the risk?
Well, Marcus' presentation is really interesting because, as he said, the Swiss have gone into regulating total counts, but it's a hard sell on a risk basis. I can tell you that because for having done 15 years of work in ingress during repairs, which is a big deal in terms of integrity of distribution systems, what you're looking for is not a small increase of total direct counts or viable counts. What you're really looking for is the elimination of any contamination that you bring in during the repairs, and you can't pick that up in a flushed system with flow cytometry. In my personal opinion, you have to look for pathogens or indicators of pathogens. And that's why flow cytometry is a hard sell. It's a great tool to monitor a system to see things changing much better than any culture-based stuff. But I'm not sure it's relatable to risk.
Thank you, Michel. And to Dan and Xiaoxia, so based, for instance, on the presentation of Francesc and Ali about functionalities, maybe to Xiaoxia about calcium carbonate precipitation, I would assume that this precipitation of carbonate links to the biological processes that are happening in the tubing. Do you think that combining metagenomics and metaproteomics could help to better understand what happens in these cells that may cause calcium carbonate precipitation, or is it only a physical process, a chemical process? Xiaoxia, are you here?
Yes, I'm here. Yeah, maybe
along with my presentation, the point that I'm really trying to show is that it's not only a physical process, but this calcium precipitation behavior is beneficial to bacteria, and they have a true choice about when they want to start doing it. They only start to do it when the density reach a certain amount, which means that it's really helping. They use it as a tool to help them. But in the sense of using genomic sequencing, I think is in the nature environment, the precipitation of calcium carbonate induced by bacteria is extremely prevalent. You can find this phenomenon in every single other environment. It's just drinking water has such a low amount of organic that we have never thought that it can happen in there. That also brings the point back that in drinking water, basically you have every process that you can find in every other environment. But because the cell concentration is so low that you simply don't have all the chance to study all the processes in there. Because if you measure the nitrate or methane oxidation, you simply don't have enough biomass, or the level is simply below detection. So I think the genomic wise, maybe it will help to find other mechanisms for the calcium carbonate precipitation or iron precipitation, which as engineer, we focus on the results, but for the bacteria, they actually have many different ways to do all these jobs, actually, and it's closely related to what they want to strive for, I would say.
Maybe this can be a point that we can discuss tomorrow on systems microbiology methods. So with proteomics, if you target proteins, you have 60% of the biomass that are protein, made of protein. Yeah. Nucleic acid is 1 to 5% only.
Yeah, but David, the issue there is that nucleic acids can be amplified, not so much on the protein side of things. So PCR is very, very helpful for the drinking water, low biomass environment, whether that's amplicon sequencing or a library prep that involves PCR. This is some of the questions that we struggle with as well. Is how useful is transcriptomics in the drinking water field? For example, because the whole process of concentration is fundamentally changing the transcript profiles. So yeah.
It's a real issue. It's a real challenge. You want the answer. It's exciting information, but it's so difficult to get something that in a sequence, when you're looking at process, you want a sequence of results to draw conclusions, and you got holes all over. So
I want to add a small comment about the functionality. David, you were asking what is the most important functionality for drinking water bacteria. Unfortunately, the answer is that they please don't grow.
Oh, come on.
And then from a biofilm. Yeah, please don't grow. It could be a nice song as well for Spotify. Mark, you have a follow-up as well?
No, maybe it's mainly a comment to the Amit. For the proteome analysis of drinking water samples, you need about, say, 40 to 60 times less volume to filter than for a genome analysis, including the PCR business, etc. So,
I think is it potentially the difference in cell counts that we are seeing between disinfected and non-disinfected systems?
No, it's just the amount of volume you have to filter to get. So you need much less volume in the end, which is also for some cases easy that you're not filtering half a day.
Thanks. And maybe to close this discussion, short question to Dan as well, that relates to what also Michel mentioned about building architectures or network architectures. So the more complex the network is and the building is, the more difficult it is to understand anything in the system. You have done an above ground scheme on your presentation. And you have all these singularities, so meaning not just straight lines, but you have some angles, like 90 grade. So what is the influence of all these singularities on the biofilm development, on biological processes happening or remaining in these systems?
You see, the angles, this one, because we built the test bed based on the limited landscapes. That's why we have the turns. And the material we choose is a PVC pipe. This is more or less to simulate the some parts of the world, like Indian, probably they use a lot of PVC pipe.
Thanks. And Michelle, you had a follow-up I have not understood before.
Yeah, it's kind of depressing, isn't it? If you have a hospital with 1,500 water points, and you I was kind of telling you two next to each other, one's really positive, the other one's got nothing. You're trying to figure out all kinds of reasons why this is. I would think that yes, it is complex, and yes, it's a distribution of response, really, and you have to look at it at a system level, and to put the pressure on the system so everything lowers. The ones that were good and the ones that weren't good, and I do think that you can manage through operating the system to even out the response and lower it. It's just more of a challenge than 200 millimeter pipes when you have this forest and this jungle of small devices and pipes and in one single building. It makes it more difficult, and that's where the risk is. So we have to figure it out.
I thought that wastewater was complex, but I think I found another field. Good luck.
Lots of DNA, and you've got a system that sees little changes in loads and stuff. This is nothing is constant in a large building. Come on.
So I have a question for Michelle. I'm very curious. So there is a lot, premises plumbing, building plumbing is obviously very, very important. At the same time, we acknowledge how complex it is. And it might be that, once we quantify or once we characterize the complexity of an entire building, next day a plumber might come in, use a gasket of different material or a faucet that's completely different. So my question is should we, where are we likely to have the most impact? As in, should we focus our attention as a field on developing methods and protocols that help rapidly assess how flushing protocols are working, or try to come up with generalized principles of how a flushing protocol might work in a large building, understanding the complexity? So it's one is rather put the energy into rapid monitoring, quick monitoring, lots of samples at the same time, and the other is let's try to develop flushing protocols. So I
think the answer is both, because you have to give people an answer on how to manage their buildings. Like I was saying, this terrible shower study, now they're all zeros, and then the PCR is under 1,500. So this is really going well. So you have, and you need all these tools to understand why it worked, so we can implement it. While in the second part, you said, we need to change the next design, the new design. That's water and energy conservation plus quality, and that's a real revolution. That's what's so exciting. But we can't push the new design until we explain why the old one didn't work. And we have to give these millions of buildings some reasonable way to control quality. And flushing, it has limitations, so has chlorination or shock treatment. But at one point, you have to figure out what to do while you have new buildings built. So it's both. I'm sorry.
Thank you very much, all the speakers, for this terrific session on drinking water. So a round of applause to everybody again. And also a round of applause to all the speakers that spoke today across all the three presentations. Thank you also for those online. And I would like to convey thank you, all the apologies for the techniques. So now we have some duties this evening to try to solve that, because we cannot do like that tomorrow. So we will do our best to solve this now to have a more stable platform for tomorrow. And this is our homework. So we will make the take-home messages of today, tomorrow morning in the opening sessions with Niti, but I think a very nice word was given by Xiao Xia that we should keep, it's please don't grow. I don't know if it's valid for all the fields, but at least from the drinking water field. Please don't grow. So with this, I suggest that you can, people who would like to go to the poster sessions, there is the evening poster sessions, and there are our colleagues from America, Canada, North and South America, Central America, who have also some posters. You can bridge to them now. And else, I wish you an excellent continuation of day, end of day and night, and we see us each other tomorrow morning, and hopefully with a stable platform. Thank you so much, and have a nice continuation.
Bye. Thank you.